All right, in this section of our notes, we're going to be talking about how mutations can lead to a change in the protein structure now that we have a better understanding of how the DNA and its bases code for the structure of a protein. So we mentioned before that a mutation is a change in the DNA, but really it's a change in the base sequence within a gene in a DNA molecule. So what we mean by a base sequence is simply this. Here's my DNA. Remember, it's double-stranded. We have hydrogen bonds between the bases. If something happens and the DNA in this area no longer reads like this, it reads like this, then that means that there has been a change in our DNA. So right here, that was a mutation. We have a change in the base sequence. So now that we understand how the bases can code for proteins, so remember we've got DNA, it's got bases on it. The DNA gets transcribed into mRNA. So the mRNA that would be produced off of CTT would read GAA. And if you're reading the codons, you can see here we're building, this is a protein, this is hemoglobin, it ends in IN. And so we would have these amino acids. In that second spot right there, you would have glutamic acid. Let me hit autofocus here. This looks a little out of focus. All right. So if our DNA reads this, then this is the amino acids that would be put in the order. This is what our hemoglobin looks like. And hemoglobin is found in red blood cells. So let's go ahead and just put HB. I know it's kind of hard for you to see on mine, but just write HB because that's what the hemoglobin would look like. And the job of hemoglobin is to bind to oxygen and to then release it eventually when those red blood cells get to tissues that need oxygen. So let's say there's been a mutation in the gene that codes for hemoglobin, and instead of it being CTT, now it reads CAT. Our mRNA would be GUA, and that codon's different. It doesn't code for glutamic acid, GLU, that amino acid. It codes for valine to be put in our hemoglobin in this position. And so then when we build our hemoglobin, it does not look like it should. It looks like that, okay? As a result, that hemoglobin in our red blood cells, it actually makes our red blood cells look like this. The red blood cells have this sickle shape. And they don't function very well. They don't deliver oxygen very well to our body tissues if the red blood cells look like that. And it's all because the hemoglobin inside of it has a different shape, and it makes the red blood cells shape like that, okay? So you can see here, if we don't have our bases in the correct order, we're not going to get the right amino acids, our proteins aren't going to be built right, our cells aren't going to be able to function. And technically you have a genetic condition if this is what's happening in your cells. Now remember, anything that can mutate DNA is called a mutagen. And I probably should have put this on the next page, but because that's what the focus of this next page is. Are the things that are mutagens. We've already talked about it before. So when you look at this diagram right here, hopefully you can tell that this is DNA replication. DNA replication. You can tell because we're making 2 molecules of DNA from one. And it's showing you that as the DNA is built, right here, something has been put in wrong. A base has been put on wrong. So we have a mutation in our DNA. So replication errors or mistakes in replication can be one possible cause of mutations in cells. We said that there's chemicals that can react with the DNA and change the structure of it. Viruses can insert their DNA in the middle of our DNA molecules, and that would then change the order of the bases. Radiation, such as X-rays, ultraviolet rays, those are examples of radiation that have a lot of energy and they can mutate the DNA. And then we also mentioned high heat. So heat can break these bonds that are holding these different parts together in our DNA molecule. And This one's showing you like heat is breaking those hydrogen bonds right there. But anyway, we've kind of changed the shape of DNA. So high heat can be considered a mutagen because it can lead to changes in the structure of DNA. Now, once we change those bases, then we are going to change the order of the amino acids in our protein. Now, it's really important that we have the right amino acids in a protein because these amino acids, this chain here, with certain amino acids in this chain, is going to cause this protein to fold up and have a correct shape. So this is a correct shape that this chain should fold up into, and this protein will work. It should be able to do its job if it's built correctly. The lights here. All right. So what this is showing you, here's the DNA, here's the mRNA, and these are the amino acids, and then eventually these should fold up and form a protein. So now let's take a look at what this protein would look like if we change one little base. So in our second picture down here, it looks like the DNA is completely the same, but right here, there should be an A. So it's going to put a star here where this mutation is happening. And instead, this has been changed to a C. Now, if you look, when the cell goes to build the protein, it looks like it's going to build it almost the exact same way until we get right here. So we've got one amino acid that's different. Even all these other ones are still the same. But that change in one amino acid right there makes this protein fold up differently. So this is the wrong shape. And many times that protein won't be able to do the job that it's supposed to do, because to do that job, it has to have that shape. So usually they won't work. Even if sometimes we put the wrong, just one amino acid different in that chain can affect the structure and the function of that protein. Okay. Now, if we have just one cell in our body has a mutation and a protein can't function, that could result in a cancerous cell. I mean, that cell could die, or it could become a cancerous cell. And if you inherited DNA that has the wrong code in it, and so therefore this code is in all of your cells, then that means that you've been born with a genetic condition. So it's not just one cell that's affected, it's all your cells that have to make this protein. This is how they're going to make it, and it's not going to function, and so you have a genetic condition. So since we're talking about mutations, that mutation that we just looked at on the previous page was a substitution mutation. And so there's 3 types of point mutations. Substitution is one we just looked at. Then we have insertion. And we have deletion. Those are the 3 types of point mutations. And so what we mean by a point mutation is a base or a few bases. Or let's just do a base. We'll put S's there in parentheses, is changed. That would be considered a point mutation. Just a change in a base or 2 or 3, just a few of them, that's called a point mutation. All right, so whenever you are asked to figure out what type of mutation is happening to DNA, well, I have to give you the normal DNA so you can figure out what's been changed. So this is showing you, this is a normal DNA sequence. Here's the mRNA. This should all make sense to you. And those are the amino acids. And then again, that whole thing is we've got a protein. So that's one protein. Protein should have these 4 amino acids in it, okay? And so when you look down here, it's saying that the substitution mutation has occurred. And so we're going to find where that change has happened. So these still look the same. ATG still looks the same. Something's going on right here. And it looks like these are still the same. So it's really just somewhere in these 3 bases we've had a change, and it looks like it's just right here. So out went the C, and in its place went a T. So substitution, I'm not adding any extra bases. I still have 12. We just take one out and substitute in the wrong one, so that's called a substitution mutation. Notice that we've only changed 1 amino acid. So we still get a protein that is mostly the same structure, but we've got 1 amino acid different in there. So in a substitution mutation, we only get usually a change in about 1 amino acid. Now, something that happens in a substitution is this protein might work. So let's just indicate it might work. It might fold up and have a similar shape and still be able to work, but it also might not work. It really just depends on what amino acid has been changed as to whether it's going to fold and it's going to be able to function. All right, now the other 2 are more severe mutations, so insertion and deletion. So notice that in this situation, I'm going to go back and show you. You see this CTT right here? Well, it's still there, but it's right here, CTT. And notice up here, it was all the part of 1 codon, and now those 3 bases are split between 2 different codons. So it looks like really the CTT, it's still there, it's just been shoved down a spot because of an insertion point mutation. So what that means is now we've just added a completely extra set of bases right there. Notice what's happening. That amino acid's different. The next amino acid's going to be different. The next one, everything from this point on is now going to be different because all of our codons have been altered if we have put in 1 extra base and shifted all of these bases down 1 position. Now, whenever that happens, when we add and then all the bases are shifted and it messes up our codon frames, this is called frame shift. So all the amino acids from that point down are going to be different. Our protein won't function. It won't work. If you change a lot of amino acids, the protein isn't going to fold and have this shape that it needs to do a certain job for your cell or your body. Okay, deletion mutation, again, you should be able to recognize that we have ATG, CCTs. Let's go down here. I have a TG, CTT. So it looks like we're missing, there should have been one here that has A. So it was deleted. So that is a deletion mutation. Notice that my T and my G, let's go back up here. They're going to be shifted down. All these are going to be shifted down 1 spot. So every codon now is going to be different. And so we say that deletion also causes frame shift. We are changing many things about many amino acids in these proteins. That protein won't fold correctly and won't work. Okay? So out of all the mutations, substitution is usually the one that creates the least change in a protein. That's not always the case, as we're going to see in an example we're going to do here. But insertion and deletion usually causes major changes to the protein structure and will result in a non-functioning protein. That's not a good thing. All right, so let's move on to this back page right here, and let's do this quick practice. And then I'm going to have you kind of do some problems and analyze what type of point mutation is occurring here. So we should be able to quickly translate our mRNA and figure out the amino acid sequence. So this is showing you, this is DNA, here's our mRNA, and we should be getting a protein that has methionine. If you look up UCA, that codes for serine. And AGU codes for serine. And then UAA is a stop. So really, our little make-believe protein here is 3 amino acids long. So this is what should be built to get a protein that will do a certain job for the cell. So let's take a look here. And it says, what type of point mutation has occurred? So if we take a look at our DNA, I've got my TAC, it's right there. And then I have an AGT is what we should be. And I do have, it looks like right here, if I change this to the G, but let's make sure that AGT and then TCA. Okay, TCA is still right here. So it looks like this isn't an insertion or anything like that. The T is being put out, and, the G was taken out, a T was put in its spot, so that would be a substitution mutation. It's the type of point mutation. There's 3 types of point mutations, okay? So now we have our mRNA. A goes with U, T goes with A. And this is showing you, remember I just said substitution usually doesn't change your proteins that much? Well, this one's going to. So if you write in our amino acids, AUG codes for MET, UAA is a stop. So we should have serine, and then we should have another serine, and then we should have a stop. Well, we have made several changes, more than just one change here in our amino acid sequence. So frame shift didn't occur, but this protein, even as a substitution, it's probably not going to function right, because when many amino acids are changed, so when many amino acids are altered or changed, a protein can't fold into a correct shape to function. Okay, now you can go ahead and cross through missense and nonsense. I'm not going to ask you those questions, and we'll talk about a silent mutation. A silent mutation is when we get a mutation, but we still actually build the exact same protein. So there was no silent mutation on this one. All right, so in example 2, we have a DNA sequence. It's telling you this is the normal DNA sequence, and then this is what we got here. So it kind of looks like we have an extra base. So that must mean that there was an insertion. We just have to figure out GAC, TCC, C. So I still have my CCCs right here. They're just been, now they're in 2 different codons. We put in an extra T. So that'd be an insertion. Anytime there's an insertion or deletion of 1 or 2 bases, yep, frame shift occurs, because look at now, the CCCs, they've been shifted down. And so now our CCCs are in 2 codon frames and not all together in 1. So that's what it means with frame shift. So yes, it occurred. We got no, it was definitely not a silent mutation. Is this protein going to function? Well, if we look here, no, we're changing every single amino acid from that point on. So let's put no, and then we can just kind of, to save time, it's the same thing. When you change many amino acids, your protein won't be able to fold correctly and be able to function. All right, last thing we're going to look at here is sometimes, let's talk about those silent mutations. So sometimes we can get a change in the DNA and we get lucky. Our protein is still going to be built correctly. So it's going to be able to function. And sometimes we get these silent mutations because of something that's called the wobble effect. The wobble effect is a good thing. So it benefits us. It sounds like it wouldn't be a good thing because if something's wobbly, that usually means like it's broken or it's not working well, right? But the wobble effect is actually a good thing. So this is called the wobble effect because sometimes you can wobble and change the 3rd base. So sometimes that 3rd base in a codon can wobble to a different base. And you still will code for the same amino acid. You've probably noticed that on your codon charts, there's actually several codons that'll code for an amino acid. So for example, if you look over here, like threonine. By the way, this is the codon wheel that's on your your biology EOC, your state test. So we want to make sure you know how to use it. You start on the inside and you work your way out. So if you're trying to find like ACG, you start A and then C and then G, codes for threonine. Okay. So notice that if you have a codon that says ACG, then threonine is going to be put in that spot in the protein. If it says ACC, you're still going to get 3 and 9. If that codon, if the DNA's changed and the codon still says ACA, you're still going to get 3 and 9. So this is an example of the wobble effect. Lots of times you can change the third base in a codon to a different base, and yet you'll still get the same amino acid. That's not always the case. Like, for example, AUG, if you change the third base and now the mRNA is AUC, you're going to get a different amino acid, okay? So it doesn't happen for all of them, but it does happen for many of the codons and their amino acids. So if you have DNA, so let's indicate this is DNA, and the DNA says GGA, all right? Well, the mRNA, the codon, so let's write mRNA codon, would be CCU. If we look up CCU, we get proline. So let's put proline in the box. Those are the amino acids. That's an amino acid. Let's just kind of label it to make sure we understand. Oops, sorry. To make sure we understand proline. All right. So if the DNA in that spot gets mutated, so let's say it's no longer GGA. Let's say it's GGG. So then the mRNA that's going to be made will read CCC in that location. But if you look up CCC, you still will put proline in that spot. And I can keep going. The DNA, if it's mutated to GGT instead of GGA, you get CCA as the codon. If you look up CCA, still going to put in proline. And then finally, the last one. If it gets mutated instead of GGA, it says GGC. Our codon is going to be CCG, and it still codes for proline. So the wobble effect's a good thing. We could get mutations in our DNA, but yet our protein is still going to be made the same way. So the wobble effect leads to silent mutations. So some mutations are less severe than others.